Greetings from Everyday GMP. I am your presenter from Everyday GMP. This is series of video. We will discuss all the definitions and term used in EU guidelines for good manufacturing practice for medicinal products for human and veterinary use. In this video we will cover terms starts from alphabet D, A, F. Further series will be covered in other videos. Total 6 videos are in this series. Series of definitions. Video number 3. Link of previous video covering A, B, and C as in description. Keep watching all videos, so let's start. EU GMP Guidelines Annex 1 Series of Definitions. Video number 3. Term starts with alphabet D, A, F. In the world of pharmaceuticals, precision and adherence to regulations are paramount. Understanding the key definitions and terminology laid out in the European Union Good Manufacturing Practice Guidelines, Annex 1, is not just important, it's essential. Join us on a journey through the intricacies of pharmaceutical manufacturing as we dive deep into the terminology that shapes this industry. In this comprehensive video series, we will demystify the definitions used in the EU GMP Guidelines Annex 1, breaking down complex concepts into easily digestible explanations. Exploring pharmaceutical definitions from EU GMP Guidelines Annex 1. Unlock the language of pharmaceuticals and dive into the core terminology that shapes the industry. Join us on this educational journey to understand the key definitions that ensure precision and quality in pharmaceutical manufacturing. Subscribe and let's begin. So before start, subscribe the channel and press the bell icon. So let's start. D value. The value of a parameter of sterilization, duration or absorbed dose, required to reduce the number of viable organisms to 10% of the original number. Dead leg. Length of non-circulating pipe, where fluid may remain static, that is greater than 3 internal pipe diameters. Decommission, when a process, equipment or clean room are closed and they will not be used again. Decontamination, the overall process of removal or reduction of any contaminants, chemical, waste, residue or microorganisms, from an area, object, or person. The method of decontamination used, e.g. cleaning, disinfection, sterilization, should be chosen and validated to achieve a level of cleanliness appropriate to the intended use of the item decontaminated. See also biodecontamination. Depyrogenation, a process designed to remove or inactivate pyrogenic material, e.g. endotoxin, to a specified minimum quantity. Disinfection, the process by which the reduction of the number of microorganisms is achieved by the irreversible action of a product on their structure or metabolism, to a level deemed to be appropriate for a defined purpose. Endotoxin, a pyrogenic product, i.e. lipopolysaccharide, present in the gram-negative bacterial cell wall. Endotoxin can lead to reactions in patients receiving injections ranging from fever to death. Equilibration time, period which elapses between the attainment of the sterilization temperature at the reference measurement point and the attainment of the sterilization temperature at all points within the load. Extractables, chemical entities that migrate from the surface of the process equipment, exposed to an appropriate solvent at extreme conditions, into the product or material being processed. First air. refers to filtered air that has not been interrupted prior to contacting exposed product and product contact surfaces with the potential to add contamination to the air prior to reaching the critical zone. Filter integrity test, a test to confirm that a filter, product, gas or HVAC filter, retain their retentive properties and have not been damaged during handling, installation or processing. Form Fill Seal, FFS, an automated filling process, typically used for terminally sterilized products, which constructs the primary container out of a continuous flat roll of packaging film while simultaneously filling the formed container with product and sealing the filled containers in a continuous process. FFS processes may utilize a single web system, where a single flat roll of film is wrapped around itself to form a cavity, or a dual web system, where two flat rolls of film are brought together to form a cavity, often with the aid of vacuum molds or pressurized gases. The formed cavity is filled, sealed and cut into sections. Films typically consist of a polymeric material, polymeric coated foil or other suitable material. That's all for today's video on terms and definitions used in pharmaceuticals. If you found this information helpful, give us a thumbs up, subscribe to our channel, and hit the notification bell to stay updated on more job, immigration and educational content. If you have any questions or want to share your journey, leave a comment below. Until next time, keep dreaming big and reaching for your goals.